A pleasant good day to everyone and welcome to Biology 171, Structure and Function. And in this recording of the presentation, Molecules of Life, we will be looking at the following. So this is just a slide which shows the flow of the work in terms of that which matches up to your course outline. So in terms of things needed, we have in terms of one, definition of the term element and list the principal elements of the body. Secondly, define the term atom, describe its structure, look at isotopes and describe their importance, the types of ions and how they are formed, differentiate among or tell the difference between molecules, compounds, and solutions. Look briefly at the following bonds: ionic, non-polar, covalent, and polar covalent. Right, then there'll be a bit of a break, and then we'll go on to discuss the importance of enzymes, the chemical reactions, describe the three major types of chemical reactions, namely synthesis type reaction, decomposition, and also exchange. And again, we'll look at explain the importance of water. Water is something that is critical to life, and we'll be looking at it and also mentioning the role that water and salts play as it relates to homeostasis or maintenance of a constant internal environment within our bodies. Then we'll be looking at we'll be comparing and contrasting acid and bases, defining the term pH, and explain the role of buffers in homeostasis. Compare the general structure and metabolic importance of the following molecules: carbohydrates, lipids, proteins, nucleic acids, and then of course our good favorite from energy, adenosine triphosphate, better known as ATP. Okay, so in terms of when we're looking at it from a chemical perspective, chemistry is the science that deals with the composition and properties of matter. It is used to understand normal and abnormal functioning of the human body. When we talk about elements, these are the components, the building blocks of all matter. And there are 92 naturally occurring elements in nature, and they are identified by names or symbols, and oftentimes the abbreviations there are modern or Latin names. And one of the abbreviations, true story, that comes to mind, of course, is the abbreviation for silver, which is AG. And one of the reasons why it is called AG is an abbreviation for Argentinium. Argentina is one of the, or was one of the places which has a lot of silver mines. So one of the things that they did in terms of when they were naming the element silver is they did it, in respect to the fact that Argentina has so many mines and produces so much silver. So they actually paid homage, as it were, to Argentina and used AG for the symbol for silver. Back to the elements. It's identified by number and is described and organized in what is known as a periodic table. For our purposes, we don't have to get into the periodic table, just know its existence. Now, atoms. So we talk about elements. Now we move to atoms, and these are the subunits of elements. So you have elements, right? The elements are found in the periodic table, and now the subunits of these are the atoms. They're the smallest complete units of matter, and they cannot be broken down or changed by ordinary chemical and physical means. So we talk about the atoms, we spoke about elements. Now let's look at the structure of an atom. When we talk about the structure of an atom, within the center of the atom is what is known as the nucleus. Very similar to a cell. When you look at a cell, you have the nucleus of the cell, which is towards the center. Within the nucleus, what do you have? You have protons and neutrons, and then revolving around this nucleus, you have electrons. Very important to note of the charges associated with these particles. Protons have a positive charge, electrons have a negative charge, whereas neutrons, they have no charges at all. So it's always very important, to take note of these things. Neutrons, no charges. Protons, positive charge as shown here, whereas the electrons, these have a negative charge. So here's a representation of the oxygen atom. And again, here you see depicted towards the center of the atom is the nucleus, and the nucleus has protons and neutrons being present. revolving around the nucleus, we see these electrons. And they all move in a particular orbitals, if you notice, or they have a certain area. Let me just change the color. So they have a specific area in which they go around the nucleus. So they revolve around it. So it's very important in that regard to actually take note. Nucleus contains the protons and neutrons and the orbiting electrons. These are the negatively charged particles. So let's talk about molecules and compounds. Molecules are formed when two or more atoms unite on the basis of their electron structures. One of the things, even at the level of molecules, one of the things and compounds, you have to recognize stability is the key. Everything wants to move to a state where everything is stable. So when we are looking at molecules, one of the things they do, they form combinations with different elements, with different substances, such that they move towards this state of being at rest, right? So very important there in terms of being at rest, in terms of having a charge which adds stability to the structure. That's very important. Compounds, these are composed of two or more elements, and the smallest subunits of the compounds are the molecules. All right, so let's do a little quick revision here, if you will. What is the smallest unit of a compound? Is it atom, proton, or molecule? Very good. The answer is indeed molecule. Very good. What is the most abundant compound in the body? Well, you'll be hard pressed to find. What does come to mind, of course, two things: salt and water. Very important there. Let's go forward. So mixtures. We're speaking about solutions and suspensions. Now, solutions, in terms of mixtures, we're talking liquids here. Solution components, they're indistinguishable from one another, and they're evenly distributed throughout the liquid's structure. Whereas suspensions, they separate from solvent. What comes to mind with suspension? What is that thing when you have, let's say, Lota on your skin and you go to the drugstore? Or do they give you that pink substance? Very good, calamine lotion. And we all know if you let calamine lotion stand on its own, let's say overnight or otherwise, when you get up in the morning, you'd see this separation. And when you see a substance separate, that shows then that there are components which are suspended in it. So the separation or the suspension, it separates one component from the other. So it's very important when you think about suspension. When you leave a suspension to stand, you usually see separation occurring. All right, both solutions are types of mixtures. What are the difference between them? Mixtures, homogeneous substances, so you don't see a separation, but most critically, when you look at suspensions, if left standing, they do separate. All right, what term describes substances that dissolve in water? Of course, we all know hydro and philic. This part here, philic, means loving. So when you look at, for instance, in the United States, there's that state, Philadelphia. Well, actually not a state, sorry, it's a city of brotherly love, right? Philadelphia. So phila, that's from the Greek, meaning love. Very important there. All right, so chemical bonds. We looked at elements, we also looked at mixtures, and we also looked at compounds. Now we're looking at the bonds, or those interactions between the atoms themselves. Let's have a look and see what bonds do exist. First of all, there's ionic bonding. And for ionic bonding to occur, we are looking at this example in which we are looking at sodium and we're looking at chlorine. So sodium and chlorine, sodium on the periodic table, its structure shows that it has a 2-8-1 arrangement. So in terms of getting stability, what it really wants to do is to lose an electron. If it loses an electron, now it has stability because its outer shell, it's now full. All that being said, sodium, in order for sodium to attain stability, it loses an electron, right? So when you lose an electron, you're losing a negative charge. And if you remember from math, when you take away a negative quantity, you get a positive, it increases. So when sodium which has 11 electrons, now loses one, it gains a positive charge. So it moves from the 281 configuration, 2 electrons in the innermost shell, 8 in the next shell, 1 in the outermost. It loses that electron, so now it goes to a 2-8 configuration, which is stable. Chlorine, 17 protons. Chlorine has the arrangement 2, 8, 7. So chlorine wants to take in an electron to attain stability. Remember we mentioned these elements want to be stable. So in other words, it could now, when you look at sodium wanting to lose an electron, chlorine wants to gain one. So what happens? Sodium loses its electron, whereas the chlorine gains an electron. So it moves from being a chlorine atom to a chloride ion. This negative charge shows that it has an extra electron in its outermost shell. But one of the important things here, it gives it stability. It now has a 2-8-8 configuration, or all of its electron shells are filled, so it is very stable. In getting this stability and accepting electron, it now has a negative charge, whereas sodium, in losing its electron, has a positive charge. When you have charges, opposite charges, positive and negative, interacting, what do you have? You have an electrostatic force here, an attractive force between a positive charge and a negative charge. So in terms of sodium chloride, the bonding that exists between the sodium ion and the chloride ion, this is known as ionic bonding. So these bonds, electrons are transferred between atoms and between atoms, and they form these ionic bonds, as we show in the example between sodium and the sodium ion and the chloride ion. Now electrolytes, these are bonds, ionic bonds form compounds that release ions when they are in solution. So one of the things with salt, the sodium chloride, when it is in solution, you have the ions being released, sodium ions and the chloride ions, right? So very importantly, and why they're called electrolytes, electrolytes is just a term which really means it carries charges. So within the liquid itself, it has sodium ions, chloride ions. The sodium ions carry a positive charge. The chloride ions carry a negative charge. And therefore, sodium chloride in solution is an electrolyte because it does have charged particles. And because of that, they could also conduct an electric current. When you think about a current is just a flow of charges. So therefore, because of the fact that the electrolytes contain these charged particles, they can conduct an electric current because of the fact these electrons can move freely within the solution. All right, what happens when an electrolyte goes into solution? The ions are formed. Let's look at another one. We looked at ionic bonding involving transfer of electrons. Now covalent, this is a sharing of electrons. So you have two types, non-polar and a polar. Depending on the partners making the bond, if one partner is stronger, therefore the sharing of the electron will spend more of its time around the stronger partner. Whereas in a non-polar covalent bond, what you'll have it usually occurs when you have two of the same elements forming this bond. So therefore, when they do form the bond, in terms of where the electron will spend most of its time, it will spend its time equally between the two elements forming the bond. So therefore, with a non-polar covalent bond, there's no net charge. But with a polar covalent, because one of the substances forming the bond is more electronegative or is stronger, it keeps the electron. The electron spends most of its time around that particular element. So therefore, what happens? You'll find that because the electron is mostly over here, this part of the combined structure will have a negative charge associated with it, and on this side it'll have a positive. When you have this separation of charge, that is known as polarity, and the type of bond is known as a polar covalent bond. This hydrogen molecule, this is an example of a non-polar because they are both equal. So therefore, in terms of the electrons, it will be shared equally between both of these elements. Right? Covalent bond, sharing of electron, and again, why do they share electron? To attain stability. All right, so we have reached this part here. Let's just do a quick recap. In terms, we looked at element and we listed the principal elements within the body itself. We defined the term atom and described the structure. We didn't look at isotopes, or we talk more about that in just a little while. We mentioned the types of ions and how they are formed, and tell the difference between molecule, compounds, and a solution, and discuss briefly the following chemical bonds. We spoke about ionic, non-polar covalent, and polar covalent bonds. All right, now let's look at the importance of enzymes to chemical reactions. Let's go. Before we look at the enzyme, let's have a look at water. Water is very important to the body. It's the most abundant compound in the body and is critical to all physiologic processes in body tissues. Deficiency of water, as we're well aware, can threaten health, and this is very important, therefore, to remain hydrated. Water is also known as a universal solvent. Things can dissolve in water. Most things dissolve in water, and it's a stable liquid at ordinary temperatures, and it also participates in chemical reactions in the body itself. We mentioned elements. Let's have a look now at compounds, particularly acids, bases, and salts. Compounds are chemical substances with specific properties. An acid, also known as a hydrogen donor, it can give hydrogen ions, whereas a base accepts the hydrogen ions. An acid, hydrogen donor, base, hydrogen acceptor. And very interestingly, there's a unique substance that is formed when an acid and a base reacts. When an acid and a base reacts, it forms a salt and water only. So very importantly, salts is formed by a reaction between acids and bases. Very important to know that type of reaction. So how do we measure the strength of an acid? Well, this strength of an acid is actually measured on what is known as a pH scale. Generally speaking, body fluids are usually between 7.35 to 7.45 on this pH scale. And this pH scale goes from 0 to 14, 0 being the most acidic and 14 being the most basic. Let's have a look at this scale. So here is the pH scale. 14, increasing basicity or alkalinity. So therefore, a substance having a pH of 14, this will be the extrema of the base scale, and something having a pH very close to 0, this would be the extrema. This would be a very strong acid. So therefore, on this, going away from the neutral point, when you go closer towards 0, the strength of the acid increases. When you move away from 7, go towards 14, the strength of the base increases. Let's have a look at some of these substances which are often found on the pH scale. Okay, so what do we have? Coffee. It is acidic, yes, has a pH of 5 on the scale. Tomato juice, 4.2. Apple juice, 3.0. Colas, yes, or Coca-Cola, Pepsis, and other black colas, they are particularly acidic, 2.5, which is why I suppose you will see it on YouTube. Coca-Cola and other colas are often used as cleaning agents. Lemon juice, yep. 2.0 citric acid is very acidic, so it's not a good idea really to suck a lemon because it does introduce a certain amount of acidity into your stomach, a greater amount of acidity. Stomach secretions in and of themselves are acidic, they're 1.5. All right, so this is the acid side of the pH scale. 7 again is neutral. Human blood has a pH of close to 7.4. And on the other side of the scale, sodium bicarbonate 8.4, bleach 9.5, lye 13. So therefore, acid going towards 0, base going towards 14. The acidity depends on what? And again, as we mentioned, hydrogen ions. That is very important on the scale. All right. Buffers. Now, what are buffers? Now, these are chemicals that prevent sharp changes in hydrogen ion concentration and maintain relatively constant pH in body fluids. So buffers are very important, and within our bloods, we do have buffers being present because, as we saw just now, the pH of the blood, it goes between 7.4, 7.45. It is very important that it remains in that region and doesn't escape or go away from it. because when it does, it could have disastrous consequences for that individual. So in order to maintain that pH, there are these buffers within the blood, and these are chemicals that prevent any sharp changes in that hydrogen ion concentration. All right, so we're almost there. All right, let's have a look at now at the chemistry of the living matter. When we look at the human body, it contains 26, the living matter usually contains 26 of 92 natural elements. 96% of our body weight has 4 elements. 4% has 9, and 0.1% contains the other 13 elements. And the major ones are carbon, hydrogen, and oxygen. Now, some of these compounds that contain this carbon, hydrogen, and oxygen include carbohydrates, lipids, and proteins. Let's just see what their structures look like. We'll look at that in a little bit. True or false? The three main organic are carbohydrates, proteins, and amino acids. Not so, right? Proteins and lipids, amino acids, those are the building blocks of proteins, right? So what the answer we're looking for was lipids in this case. Let's go forward. Carbohydrates, they usually consist of monosaccharides or the simple sugars, right? An example is glucose. Examples of carbohydrates. You have the disaccharides and the polysaccharides. Polysaccharides are the long chain, long chain one, namely starch and glycogen. So these are examples diagrammatically of how they look. Up here we see glucose, example is dextrose, right? It's a monosaccharide because it has one unit making it up. When we look at sucrose, which is table sugar, it's a disaccharide because it consists of 2 components. Then you have these poly meaning many, consists of many repeating units, and one such example is glycogen. All right, so we looked at sugars, carbohydrates, and now let's look at the lipids. Example of lipids are triglycerides, phospholipids, and steroids. Steroids are very important in our body, and they exist as cholesterol, steroid hormones, and of course the sex hormones. Very important. Biologically, we must always remember the reason we are here is to reproduce. If we do not reproduce, we will disappear as a human entity, as a species. So it's very important. So therefore, when you're looking at the sex hormones, very important, and you will look at that later on in SNF2, Structure and Function Part 2, to look at these hormones and their compositions. So let's have a look. Here we're looking at lipids. One of the things you will notice in terms of the lipids, this is a simple fat here, triglyceride, right, is composed of fatty acids and glycerol, right? So the glycerol entity is at the end, and here we have these fatty acid chains, attached to the glycerol. Here's another example here. Cholesterol, which is a steroid, showing its structure. So it's important to note the structures, because if you do see them, you will be able to recognize them in terms of, let's say, in an assessment. And for the purpose of this course outline, the structure recognition is important for this course. Carbohydrates, we look at lipids and steroids. Now let's have a look at proteins. As we mentioned previously, the building blocks of proteins are amino acids. Proteins also consist of enzymes. Enzymes are proteins which catalyze or alter the rate of reactions. So very important, they're catalysts, and they also serve, these proteins serve as substrates. Substrates meaning something could bind or attach to it, and then they could initiate a change. These substrates are often found in the membranes of cells, and when you do have binding, it causes a conformational change. Conformational changes as it relates to proteins within membranes, this is very important when you're looking at gates. And later on, when you look at in terms of neuroscience and your nerves, when you're looking at the generation of action potential and how it is the action potential actually crosses the synapse, you would recognize that you have this changing of this electrical signal into a chemical signal. Chemical signal then moves across the synapse, then it binds at specific receptors or at specific areas. And when they do bind at these areas, you have conformational change, and then you have the repropagation of the action potential at a site distal to the original one. So here we are looking at, in terms of proteins, and important to note, proteins have different structures or conformations of how they are arranged. But first, let's take a step back. Simple amino acid, and they're called amino acids for a very simple reason. These are the building blocks of the proteins. Amino acids because they have an amino group, an NH2 group on one side, and an acid group, C double bond OOH, right? So this is known as an acid group. This is an amino, so therefore put them together, you get amino acid. And this is an example of a simple amino acid. Notwithstanding these amino acids, when they join together, they now form proteins. And in terms of the structure of these proteins, they could be coiled, pleated, or they could aggregate. This looks very much like hemoglobin. They could aggregate to form a folded structure. Right, so this is just showing enzyme. I did mention that how, in terms of binding of proteins at the level of a receptor, could cause conformational change, analogous to binding of an enzyme to a substrate. So here we have the binding of the enzyme, the substrate binds to the enzyme, and what does it do? It produces a conformational change in terms of bringing these two entities, two of the substrates together to form a final product. So this is very important where that is concerned. All right, so that is the end of the presentation thus far. And what did we do? Well, we looked at different things. We looked at the elements, we looked at molecules, we looked at the different bonds which exist, ionic bonds, covalent bonds. We moved on, and then we are looking at pH, measure of acid, right? In terms of on the pH scale, 7 is neutral. You move towards 1, you're getting acidic, and then you move towards 14, you're getting greater basicity, or more basic, or an increase in basicity. Very important there. So after that, we moved on, and we just looked at the last topic there, in which we are looking at the structures of carbohydrates, proteins, and fatty acids. That was very important as well. So there is this represents the end of the presentation, and so we'll close off here, and you could review at your leisure and then answer the quiz that follows. All right. Hope you enjoyed it. Have a great one. Bye-bye.